Okay, hello everybody. We'll make a start. So I'd like to start by acknowledging country, the traditional owners and custodians of the land on which we meet today, the Wurundjeri people of the Kulin Nation, and pay my respects to the elders past and present, and the elders from other communities who may be here today. So welcome everybody to the 2022 Postgraduate lecture series. I'm really excited that it's going to be on cancer, which is the area of research that I love. And we also are welcoming students from the CC PhD program over at the VCCC as well. So I'd like to start by just saying thank you to a few people that have helped us put this series together. So Rosie Falcone, Kerry Coe, Julia Mann, and Catherine McLean. They've been really helpful in getting this all together. So we're really, really excited that we're back in person after 2 years. And so I would encourage as many students as possible and postdocs and RAs to come into the Davis Auditorium and kind of really get involved in this lecture series. This is really a series for the students. So the lectures are going to be chaired by the students. Please get involved, be brave, ask a question. Chances are if you don't understand something, there'll be somebody else who also doesn't understand it. And we're going to give some incentives for students to ask questions. So if you ask a question, then your name will go into a drawer at the end of the series, and there'll be some kind of prize. So if you've asked a question and you're a student, please come down to the front and write your name down before you leave the room. So the more questions you ask, the better chance you have of winning. We're also going to bring back some traditions from Davo. So every time a student used to ask a question, he would throw a sweet or a chocolate at them. So we've got chocolates down the front, and either me or Ziwen will throw them at you. Our aims are bad, so stay alert, no one go to sleep. Okay, so as I said, this is your lecture series, so you'll be chaired by different students, so you won't hear from me again. And I'm going to pass over to Ziwen Tan, who's an undergraduate student from structural biology, and she will be the chair for today's lecture. Okay, over to you, Ziwen.
Thanks, Gemma. Hi, everyone, for attending the seminar for today. So this seminar is the first one of the entire postgraduate series on cancer, with the first one being on the introduction to cancer, given by Professor Andreas Strasser, working at the Blood Cells and Cancer Division, and also the cancer research and treatment theme at WEHI. So Andreas is a cancer researcher trained in cell biology, immunology, and molecular oncology, and has made discoveries that defects in cell death can cause cancer, autoimmune diseases, and impair the response of cancers to chemotherapy. So his current work is aiming to reach a more detailed understanding of the molecular control of programmed cell death, mainly apoptosis. His team is exploiting the knowledge to discover novel treatments for cancer and for autoimmune diseases that directly activate cell death machinery. So welcome, Andreas. Thanks for the seminar.
Thank you very much, Thuan, for your very kind introduction, and thank you to Gemma and the team for selecting me to give this introduction into cancer biology. So the purpose of my talk, the way I see it, is give you a very broad overview. You'll get fantastic lectures from many, many people, absolute world leaders in the field that are going much deeper. And I'll just want to give you a good start into the lecture theater, into this lecture series. I also plan to try to provoke you and think about stuff that you believe might be textbook knowledge, and try to question it and say, well, is that really true? Because I think a lot of the advances come from being questioning, maybe even slightly disrespectful for what you think is cut in stone. So this is the disclosure statement I have to make officially, so you can read it, and I'll get started. So the history of cancer, let's start with that. So cancer probably existed throughout human history and probably through prehistory as well for people who got old enough that they would have been vulnerable to getting cancer. And the first written description that you can see in the literature is from the so-called Edwin Smith papyrus from ancient Egypt, so 1600 years ago. So that's almost 4,000 years ago that a cancer was described. The second description came from Hippocrates, the person on who starting medical doctors make their oath to do good and only good from ancient Greece, and he described actually several types of cancers, and that actually gave rise to the name of cancer. He called them in ancient Greek carcinos, which means crab, the animal crab, not the disease crabs. The name comes from the appearance of the cut surface of a solid cancer, which, because of the blood vessels that stretch out to all sides of the cancer, actually makes it look like a crab. And you can see that also in the symbol from, for example, the Australian Cancer Research Foundation, the ACRF. So moving on, what's actually the definition of cancer? So if you go to Wikipedia, then this is what you will find. Cancer is an abnormal growth of cells which tend to proliferate in an uncontrolled way, and in some cases are able to metastasize, spread to distant sites. So yeah, this is something you find in the textbooks. Is it actually 100% true? This is my first challenge. And I like exceptions to rules, and there is at least one exception to the rules, and this is Hodgkin's lymphoma. So in Hodgkin's lymphoma, the transformed cell is called the Reed-Sternberg cell, which is this big fat cell in the middle. And it's actually an absolute minority of the cells in Hodgkin's lymphoma. The vast majority of cells are actually reactive lymphocytes and myeloid cells that make up the bulk of the tumor. So it's actually not always true what the standard textbook statements say. And Reed-Sternberg cells are actually mostly post-mitotic, so they don't actually divide. So if we are in the lab and we want to prove that a disease that we see in a mouse model, for example, which is what Gemma and I work on, or in a human is a cancer, how do we actually prove that? So in the mouse, a very commonly used test that is highly definitive is transplantability. So we work on lymphomas and leukemias. So we would take the lymphoma, take a small number of these cells, inject them into a recipient mouse, and then if the recipient mouse a few weeks later comes down with the same type of leukemia or lymphoma, that is intracontrovertible proof that the primary mouse had a tumor, a lymphoma. The problem is that if this assay doesn't come up positive, that doesn't prove that the first mouse didn't actually have a cancer. Some cancers are just not easily transplantable. And I could give you a few examples. Another test that is very often used and of course gives rise to a lot of very useful tools for our research is to put the cells from the tumor of the primary mouse in tissue culture. And if you can easily make a cell line, you can probably accept that as proof that there was a cancer in the primary mouse that you were looking at. Yeah, what about in humans? Well, in humans, you can't do transplantations, but into another human, but what you can do is histological examination, flow cytometry, karyotyping, many sorts of imaging nowadays, sequencing the tumor DNA, comparing it to the DNA from the normal cells of the patient to look at gene defects. But you can do transplantability, and in this institute this is done a lot. Of course, as recipients you will have to use mice that are highly immunodeficient, like the NOD/SCID common gamma chain mice or the short NSG mice, and this is a picture I got from François Vaillant from Jeff and Jane's lab, where they transplanted a breast cancer into an NSG mouse, and then the mouse got a tumor that, at least by histology and by FACS analysis, looked very, very similar to the tumor as it came out of the human, and therefore you can assume that it's a very faithful model and something you can do interesting experiments on. Okay, now coming to classification and the nomenclature of cancer. So cancer is not one disease, it's a very complex group of malignant diseases. There's more than 100 different types of cancers in humans alone that we know of. And they have inherent differences in underlying risk factors, different abnormalities, genetic abnormalities and epigenetic abnormalities that we know are driving them and influence their development and also their response to therapies that are available. So here's the nomenclature. So the name carcinoma is used for cancer derived from epithelial tissues like breast carcinoma or lung carcinoma. Sarcoma is the name you use for cancer that was derived from connective tissues, such as bone, for example. Lymphoma, leukemia, multiple myeloma are cancers that come from blood cells, leukocytes, or erythroid cells. Germ cell tumors are cancers that arose from pluripotent stem cells, usually in the embryo, and starting in the embryo. An example of that is testicular cancer. And blastomas are cancers derived from embryonic tissue, and a well-known example of that is so-called teratomas. So cancers that are malignant are usually called carcinoma as a suffix, and using the Latin or the Greek word to indicate the tissue of origin of that kind of carcinoma. One example would be hepatocarcinoma, which means a cancer of liver cells. Benign tumors, which are not malignant cells, are often have the name oma as a suffix. So one example would be leiomyoma, which is a benign tumor of smooth muscle cell origin. Very unfortunately, some malignant cancers have an ending of noma, which of course is very similar to oma. One well-known example is melanoma. So there's a potential confusion that it just exists and we have to live with. So even cancers of one specific tissue type, for example, breast cancer, is well known now to be highly heterogeneous between different patients. And this is histological demonstration of that. This is histology that I got slides from Jeff and Jane and Francois. So you can see an example of invasal ductal carcinoma on your left side, and then in the middle, invasive lobular carcinoma, and then Paget disease on your right side. You can also classify now with a lot of genomic data and gene expression data cancers such as breast cancers by genomic analysis or gene expression analysis. And many years ago, this has led to this currently well-accepted classification of luminal subtype A, luminal subtype B, ERB2 positive breast cancer, basal subtype, and then normal breast-like tumors. So this classification is not just for the sake of classification. It actually is incredibly important because it corresponds for patient outcomes. So as you can see here, the luminal A-type cancer patients have a much better prognosis and long-term survival compared to, for example, the ERB2 positive breast cancer patients, which have the poorest outcomes of all. So these stratifications are really, really important because they provide information to the clinician, information to the patient and their families, and also information of which type of therapy should be used upfront for a given patient. heterogeneity of cancer is not only at the level of different patients that have a cancer of a given tissue type, like breast cancer, but even within the cancer, within a single patient, we now know that there's a huge amount of heterogeneity. And some of the cells, as I tried to draw here, would have the same oncogenic mutations and maybe the same epigenetic abnormalities, and the mutations that are really important for driving this type of cancer are called the driver mutations, and the mutations that you find that you think, well, maybe they don't do much, they're called generally passenger mutations. So this heterogeneity, again, is actually has a huge amount of importance. For example, and obviously I can't spell metastasis, as I just see, when metastasis occurs from the primary side of the tumor into distant sites, it could be that cells with a particular context of mutation 1, 2, 3, and 4 might do much better, be better equipped for that than another type of tumor cell that is very good at growing in the primary site and has mutations 2, 3, 5, and 8, for example. So therefore, the profile of mutations and the profile of gene expression that you might find in the metastasis could be significantly different from what it is in the primary tumor site. Of course, the diversity of genetic mutations that are there in the primary tumor and also in the metastasis itself can have a huge impact on how the individual cells within the tumor will respond to a given therapeutic intervention. Again, it could be that one context of 5 or 6 mutations could equip the cell much better to resist being killed by DNA damaging drugs or some other kind of drugs than neighboring cells that in the absence of treatment were just as good as expanding and proliferating. Okay, move on to what are the causes of cancer. So cancer is caused by defects in genes. In a beautiful book, it was written that cancer is a malady of the genes. And these genes that are mutated in general, as I will come back to very soon, are genes whose proteins regulate cell proliferation, cell growth, metabolism, cell migration, cell survival, and many other processes. And of course, the protein products of these mutated genes can be very attractive therapeutic targets, such as the abnormal kinase BCR-ABL in Gleevec, in CML, which can be blocked by the drug Gleevec, and I'll briefly touch upon that later on. But it's not just genetic changes, it's also epigenetic changes that can have substantial impact on driving the development of a tumor in the first place, and also driving its metastasis, for example, and also its response to whatever therapy is thrown at the patient. And therefore, there is some hope that reversal of epigenetic changes by certain drugs, such as inhibitors of histone deacetylases or HDAC inhibitors, could have some promise in cancer therapy. So DNA lesions, many of them that are found in a cancer when the patient is being diagnosed, they occurred spontaneously during DNA replication. So billions of cells replicate in our bodies every day. And of course, a very large number of bases have to be copied, and there will be mistakes. Our cells are equipped with a phenomenal DNA repair machinery, so most of the mistakes will be corrected, but not always all of them. And most of these mistakes will be in an area of the genome that really doesn't matter very much, but occasionally, unfortunately, it could be a mutation that activates an oncogene or inactivates a tumor suppressor gene. And then, of course, that's the first start along the way of transformation. Then descendants of that cell with the first mutation at some stage later might acquire a second mutation, a third mutation, etc. And if the combinations of genetic and epigenetic abnormalities are the right ones for benefiting tumor growth, that's when the tumor will become apparent. DNA lesions can also be induced by environmental carcinogens. I guess smoking tobacco is a well-known one. Exposure to UV light in Australia is a very well-known cancer-inducing factor. There's toxins in food, not so much in the Western world, but there's many examples of cancers in the third world that are driven by toxins that are prevalent in food. Exposure to gamma radiation, of course, and also exposure to many anti-cancer drugs, particularly those that induce DNA damage. So a very unfortunate consequence of saving children that have acute lymphocytic leukemia, such as pre-B-ALL, is that they need treatment that is highly aggressive and involves several DNA damage-inducing drugs. And that has the inherent risk that a child that might be saved at the age of 9 years from its pre-B-ALL and will have a normal life after that might 20 years down the track get a cancer that is a direct consequence of DNA lesions that were caused by the treatment that the child was given. And of course that gives rise to a huge effort in finding the absolute minimum toxicity schedule that is given to children with cancer to avoid precisely that 20, 30 years down the track they will come down with another cancer. Of course, there are very unfortunate individuals where the first mutation putting the cell on the path to becoming a highly malignant cancer is already there in their genome, inherited from their father or from their mother, and therefore many fewer, or at least one fewer mutation is needed for a cell to go to a malignant tumor. And examples of that are mutations in the breast cancer genes BRCA1 or BRCA2, where patients are highly at risk of developing either ovarian or breast cancer. And I'm sure next week you'll hear a lot about the Li-Fraumeni syndrome, which in most cases is due to an inherited point mutation in the p53 gene, a very well-known tumor suppressor that I'll touch upon briefly later, but you'll hear a lot more from David Lane. And these patients, they are at risk of developing several different types of tumors, probably depending on other genetic factors that a particular family will have, and usually at a very, very young age. Approximately 25% of human cancers are thought to be driven by viruses, and examples of that are listed at the bottom. So certain papillomaviruses are known to cause cancer of the cervix in women. And thankfully, due to the efforts of Ian Fraser and in Queensland, there is now a vaccine against papillomavirus. It doesn't cover all of the strains, but I believe about 80–85% of strains. So this cancer hopefully will soon be eradicated from the planet. A huge success. Hepatitis B virus is a cause of liver cancer. Epstein-Barr virus that Gemma used to work a lot on and still does some work on is known to cause lymphoma in transplant recipient people that are immunosuppressed. It also is responsible for a subset of gastric cancers and in East Asia and nasopharyngeal carcinoma. Of course, some of the virus carry oncogenes, so that's, again, the virus brings in one oncogenic lesions and then additional oncogenic lesions that the cells, the infected cells, sustain will bring about the malignant cancer in due course. What are the outcomes of genetic lesions that cause the development of cancer? So generally you can subdivide them in gain-of-function mutations, so they are genetically dominant. And they increase the activity or the aberrant induction of the activity of a protein, for example, the mutated BRAF kinase that causes melanoma. or overexpression of a protein that should be expressed at much lower levels, such as MYC in Burkitt lymphoma. Occasionally, it also a mutation can cause the expression of a fusion protein, and a good example of that is BCR-ABL in CML, which I've mentioned already before. Then the second way to broadly classify the consequences of mutations is loss of function mutations. So they are genetically recessive. So generally, for them to drive tumor development, both alleles of that gene must be mutated, or one must be mutated and the other genetically lost. And this can lead to complete loss of a protein, or at least complete loss of its function. And good examples of that is retinoblastoma, which is a protein that regulates cell cycling, and its loss causes retinoblastoma in children, and P53 is an example of that as well. But then there's also a combination of loss of function and gain of novel functions ascribed to some cancer mutations, and the best example of that is P53, where one allele often carries a point mutation that actually allows the protein to be expressed, usually at very high levels, and it's thought that this protein not only has a loss of its normal function, but also a dominant negative effect, and in some cases also gain of function effect. I'm sure David Lane will expand on this substantially. Okay, so what are the nature of the oncogenic lesions in DNA, the different ones that can start the development or propagate the development of cancer? So start with point mutations. So this is change in one single base pair in a gene. This can increase or change the function of the encoded protein, such as constitutive activation of a kinase. Again, the example that I'd like to bring is point mutations in BRAF that are well known to drive melanoma. A point mutation, of course, can also cause the opposite. It can be loss of function of the encoded protein, and P53 is one example. And again, as I mentioned, these are recessive mutations, so you would need to lose or mutate both alleles to have an effect on the cell. Of course, a point mutation, if it's in a regulatory region of a gene, it can also have an effect. If it's in the promoter or the enhancer or the 3' UTR of a gene, this could change the transcriptional regulation of a gene, and therefore its encoded protein. It could either be much more of that protein made or much, much less of it made. So it could be, depending on the nature of the site of the mutation, it could be a gain of function or it could be a loss of function. Another type of DNA lesions that is seen very commonly in cancers is chromosomal translocations. They're highly prevalent in leukemias and lymphomas. And basically, what this is that a bit of one chromosome is joined to another chromosome. And then if it's a reciprocal translocation, then it means from the second chromosome, that bit that it was replaced is now going back to the first chromosome from where the donation of DNA was made. So this example here is a reciprocal chromosomal translocation between chromosome 4 and chromosome 20 in the human. So chromosomal translocations, they can cause fusion genes that encode fusion proteins. And again, a very good example of that is the BCR-ABL kinase, where essentially the Abelson kinase, instead of being just a kinase protein, is now fused to the BCR protein, and that causes substantial misregulation and hyperactivation of the Abelson kinase, driving CML, and is also found in a small number of other types of leukemias and lymphomas. Chromosomal translocation can also cause deregulated expression of a gene by that gene becoming subject to the regulation of a foreign promoter or foreign enhancer. And one example that I chose here is the overexpression of BCL2, the B-cell lymphoma gene number 2, as a result of the 1418 chromosomal translocation, which is highly prevalent in follicular lymphoma and causes abnormal overexpression of BCL2 in these cells and drives that lymphoma. Another type of chromosomal abnormality is called breakage fusion bridge cycle, where as a consequence of erosion of the telomeres, which essentially protect the ends of chromosomes, and sometimes after many, many divisions of a clone of cell can become eroded. And when that protection by the telomeres becomes defective, then you can have fusions between chromosomes, and then at the next cell division, you can have breakages, and that then during the next replication can lead to even worse abnormalities. It can lead to focal or even entire chromosomal amplifications. And yeah, as written here, so this can lead to what is called somatically acquired gene copy number amplifications, which then lead to increased expression of the corresponding protein. Examples are the anti-apoptotic protein MCL1 is probably somatically copy number amplified in about 15% of all human cancers across diverse cancer types. And BCLXL, another anti-apoptotic gene, is amplified in a much smaller fraction of human cancers. You can, of course, also have the opposite. You can have somatically acquired loss of a copy of a gene. And of course, if that occurs on both alleles, that can lead to the loss of a tumor suppressor. Now, all of these genetic abnormalities that I just described, in essence, the consequences of these abnormalities of all oncogenic mutations is that they will make, in a simplistic manner, they will make the cells that carry that abnormality do something that they really shouldn't do, that we don't really want them to do. And that could be to proliferate when they really should be quiescent and not proliferate. It can make the cells survive when really they should undergo programmed cell death. They can migrate from the tissue that they are into another tissue, and of course that can be the reason for metastasis. It can make cells be metabolically highly active when really they normally should be metabolically relatively inactive. And these so-called acquired capabilities of neoplastic cells or malignant cells have been about 25 years ago dubbed by Weinberg and Hanahan the hallmarks of cancer. It's become a really, really helpful way to look at cancer, and this is the most updated version of the hallmarks of cancer, this time only written by Doug Hanahan, that came out in Cancer Discovery just a couple of weeks ago. The earlier two versions of the hallmarks of cancer were published in Cell, and I think the first one has been cited nearly 100,000 times. So if you want to get your citations up, come up with a really exciting scheme and write it up as a review, ideally in a big journal, and your career will be made. Not that Hanahan and Weinberg needed that to make their career, just as an aside, they made some very exciting discoveries. So amongst the hallmarks of cancer, there is deregulated metabolism of cells, resisting cell death, genome instability, the ability to access the vasculature and growing the vasculature to feed a solid cancer, activation of the processes of cell invasion and metastasis, tumor-promoting inflammation, replicative immortality, that means cells can divide hundreds of times rather than have hit the so-called Hayflick limit, which is something that I'll come to in a minute, which is essentially limited ability of cells to divide a number of times and then can't divide anymore. avoiding immune destruction, evading growth suppressors, and of course sustaining cellular proliferation. Many of them are at least in their regulation and also in their mechanisms that they operate are linked with each other. But overall, this is I find it a very helpful scheme, and I'm sure many others as well. So I'll just go into some examples of now linking a particular mutation to a particular hallmark of cancer. And again, I'm not going to be able to be totally encompassing all of the hallmarks of cancer, but just a few by giving you examples. So self-sufficiency of cells to have their growth and proliferative signals. This is a typical way how surface receptors or so-called growth factor receptors or cytokine receptors function. So they become activated when a cytokine binds to the 2 receptor chains, or sometimes it's 3 receptor chains. That then activates a downstream signaling, such as here depicted by activation of the JAK kinases, and then they can activate transcription factors, in this case called STATs, that then activate gene expression and change the behavior of the cell, making it divide, making it metabolically active. So this pathway can be activated constitutively in cancer in several ways. One example is mutations in the interleukin-7 receptor, which is important for the division of early progenitors of B cells and early progenitors of T cells. And there is a fraction of human TALLs, or T-cell acute lymphocytic leukemias, that are driven by point mutations in the IL-7 receptor that essentially make it constitutively active even in the absence of the ligand IL-7. The other examples are mutations in the kinases, the JAKs. The name, actually, JAKs were discovered just about 100 meters away here in the old Ludwig Institute when it existed by Andrew Wilkes' group. And just as a funny aside, at the time the student who discovered JAKs, he was kind of quite disappointed. They were trying to find new regulators of cytokine receptor signaling, and then they found a kinase. So the name that he gave it was just another kinase. So that's what JAK means. I'm sure that he will be very happy about his discovery now, because they are absolutely a huge field in their own right. And there's now inhibitors of JAKs used in the treatment of certain cancers. So it's not just another kinase. It's a hell of a lot bigger than that. So JAKs are mutated in polycythemia vera, in idiopathic myelofibrosis, and essential thrombocytopenia. So these all belong to the myelodysplastic syndromes, which are by themselves not a very aggressive cancer, but they have a huge risk of progressing to a highly aggressive acute myeloid leukemia, and are a forerunner of that. So what about the enablement of replicative immortality? So as I indicated briefly before, so cancer cells can undergo probably, we believe, limitless numbers of divisions, whereas normal cells from humans and mice, in many cases, they're constrained by what is called the Hayflick limit. So Hayflick was the first one who in tissue culture observed that when he put human fibroblasts in cultures, they could divide. And then after about 12 or 15 divisions, that was the end, and there was a plateau of cell numbers, and these cells would never divide again unless they acquired the loss of a tumor suppressor gene or a very potent oncogene. So as I mentioned before, telomeres protect the ends of chromosomes, and one of the reasons for the limitation in numbers of cell divisions that cells can undergo is the erosion of the telomeres. So the replication of the telomeres is not perfect. So with every cell division, telomere becomes a bit shorter, and at some stage that prevents further cell division. So there are two opposing consequences of the process of telomere erosion. So on the one hand, Yes, it's tumor suppressive because it limits the growth or the further proliferation of cells, but unfortunately, as I already showed you before, loss of telomeres can cause massive genomic instability, and then can then give rise to this breakage fusion bridge cycle that I briefly described. And then that, of course, can drive the activation of oncogene, the loss of tumor suppressor genes. So it's really very much a two-edged sword. Another hallmark of cancer that, well, obviously is extremely close to people like Gemma and myself and Marco in the room and Philippe, is evasion of cell death. So cells that are undergoing neoplastic transformation experience a tremendous amount of different types of cellular stresses. For example, the supply of nutrients can be limited, the supply of growth factors can be limited, cells can sustain endoplasmic reticulum stress. If they divide at a crazy speed, there can be replication-induced DNA damage, and that causes DNA damage stress. And so many of these stresses actually induce apoptotic cell death. So, of course, if a cell that is in the early stages of transformation, if it undergoes apoptosis and dies, well, you will not have a problem from that cell. It will never become a cancer. It's dead as a dodo. So therefore, cell death is a tumor suppressive process. I'll briefly just show you the mechanisms of apoptotic cell death as we understand it. So the way it's regulated is that the effectors of cell killing in apoptosis are BAX and BAC that you can see in orange at the bottom. Once they're activated, the cell will be killed because they permeabilize the outer mitochondrial membrane, and a lot of downstream things happen that are not that interesting because the cell is already committed to being dead. In healthy cells, BAX and BAC are restrained by the pro-survival proteins, the BCL2-like pro-survival proteins, as they're labeled here in blue. And when a cell is activated to undergo apoptosis, this happens because in response to stress, a third subgroup of the BCL2 family, the BH3-only proteins, are upregulated either through transcriptional or post-transcriptional processes or both. These BH3-only proteins, they bind to the pro-survival proteins with incredibly high affinity, and that then liberates BAX and BAC to do their job and kill the cell. So not surprisingly, mutations in all of these three subgroups of the BCL2 family are actually found in human cancers. So epigenetic or genetic changes in apoptotic BH3-only proteins like BIM and PUMA are found in certain cancers, such as some kidney cancers and some lymphomas. BCL2 overexpression, as I mentioned, is a cause of follicular lymphoma because of a chromosomal translocation that I mentioned. MCL1 gene is amplified in a very large number of human cancers. And even BAX and BAC, where you have to remove both of the proteins and therefore basically 4 alleles, very rarely you can find tumors that actually have mutations in both alleles of BAX and BAC. It's been seen in colon cancers with DNA mismatch repair abnormalities, where of course the DNA abnormalities just go completely haywire. A nice example of the tumor suppressive nature of cell death is an experiment shown here that Alex Egle did when he was a postdoc with Suzanne Corey. So you can see mice that in their lymphocytes overexpress MYC are in the gray broken line. They develop tumors with the incidence shown here. If you remove just a single allele of BIM, you get a massive acceleration of lymphoma development, and removal of both alleles of BIM gives an even further acceleration. So here again, I want to try to challenge you. So the textbook knowledge is blocking cell death causes cancer. Makes sense from what I said. You have an early neoplastic cell, it dies, it doesn't cause cancer. But is that actually always the case? And this is one of the most amazing experiments that a student did in my lab, Eva Michalik, who is now at the Peter Mac. So she used a well-known mouse model of lymphoma development. So you take young mice, 4 weeks of age, and at that time, and 3 times a week later, you give it a very low dose of gamma radiation, and the mouse will develop a thymic T-cell lymphoma. So, of course, you activate the DNA damage-induced pathway to program cell death or apoptosis, which requires p53, which is a transcription factor. p53 activates the BH3-only protein expression, PUMA and NOXA. So our assumption was, okay, well, we get these tumors with low-dose radiation at a given time. We take out a barrier that induces apoptosis to tumor development. We'll get massive acceleration of lymphoma development. And I really, love these types of experiments. So this in gray is the control mice. If you remove p53, you get a slight acceleration, or one allele only, of tumor development. And we thought we take away PUMA, the mice will get lymphoma super fast. Eva said, this is really strange. These mice don't get any tumors at all. Even the heterozygous essentially don't get tumors. So that was absolutely sensational. I love these experiments. You have a preconceived idea, you think this is how it's going to work. I will bet my house on it, and then the exact opposite happens. And she worked out actually the explanation for that. So in control mice, with every low dose of radiation, a large number of mature hemopoietic cells, red blood cells, white blood cells will be killed. And a consequence for the mouse, therefore, is that its stem cells and progenitors have to work really, really hard to make new white blood cells and red blood cells. That makes these cells divide at a pretty stressful rate. And that, of course, then causes oncogenic lesions, as I mentioned. Well, in a PUMA knockout mouse, the mature leukocytes are completely protected from low-dose radiation-induced killing. So the stem cells and the progenitor cells, they don't have to work at all. They can do what they're best at, and that is just sit there and sleep and do nothing. So this really showed to us that hopefully the world as well, that sometimes things are the exact opposite. So sometimes it can actually be cell death, even by apoptosis, that can actually be absolutely required for the development of a tumor. And there's many, many other examples of that. For example, blocking cell death in the hepatocytes in the liver can prevent the development of hepatocellular carcinoma driven by RB mutations. And it can also, in models of AML, it can prevent tumor development. So evading immune destruction is another hallmark of cancer. For a long time, this has been very controversial because a lot of work done in the 60s, 70s, and 80s was really, really shoddy. Some of it now exposed as essentially scientific fraud. So that didn't give the field a very good reputations. Also, a lot of the experiments used virally transformed cancer. So, of course, if you then put that cancer into a naive mouse in a transplantation experiment, you will get an antibody and T-cell response against the viral antigens. And then, of course, you get eradication of the cells. Even if you would put in a normal cell that expresses viral antigens, it would be removed. However, there was also some good evidence there, even from human patients. There is evidence of spontaneous disappearances of cancers in humans that's been known for a long time that could not be explained, and sometimes was actually associated with a strong immune response against a viral infection, for example. And then, of course, what really nailed it is when Jim Allison discovered the negative regulators of the activation of T lymphocytes, CTLA4, and Honjo discovered PD-1, and it was shown that if you block the function of these proteins with blocking antibodies in at least certain cancers, you can actually get a huge improvement in survival and in many cases even cures. A neglected hallmark of cancer that I'd like to talk about briefly is that's not in the list of Hanahan and Weinberg yet is evasion of cell differentiation. So blood cells, for example, they all come from progenitors and stem cells, and then they commit to a lineage, and then they become mature neutrophils or white blood cells like B cells or T cells. There's a very interesting leukemia that is called acute promyelocytic leukemia, or APML, and this is driven by chromosomal translocation between the retinoic acid receptor alpha and PML, and this causes expression of a deregulated transcription factor. What is really interesting of this tumor is that when you treat these leukemias in tissue culture, as shown here with all trans retinoic acid, then these cells that are highly proliferative progenitors in nature, they all differentiate out and become mature, mostly neutrophils that have a short lifespan and then just die. And in fact, this leukemia responds extremely well to the combination of all trans retinoic acid and arsenic trioxide with 70% progression-free survival at 12 years. So I think this is a clear example that evasion of cell differentiation should become a recognized hallmark of cancer. Another what I would call enabler of cancer development that is not much appreciated is cell competition between cells, usually progenitor cells. So this is a mechanism where some progenitors will do much better than others, perhaps because they have a growth advantage, perhaps because some progenitors kill the less successful other progenitors. And there's a really nice experimental system of that was developed by Hans Reimer Rodewald in Germany. And this is where they transplant thymine under the kidney capsule of a mouse. And if the recipient mouse has defective progenitors that can't really replace the progenitors in the grafted thymus, then you end up with a T-cell acute lymphocytic leukemia, as you can see here in the large spleen. And the reason for that is that if the progenitors that sit in the thymus, if they can't be replaced every few weeks by fresh, healthy progenitors, then the progenitors that are there already in the thymus, again, they have to work much harder. They will divide many more times. They will undergo DNA replication at a much higher rate than is healthy for them. There will be consequences, mistakes, oncogenic lesions, end product is a TALL. So I'm coming towards the end. So these, again, the hallmarks of cancers, and a lot of work by pharma companies and academia and biotech companies has brought about actually drugs that specifically target key effectors of some of the hallmarks of cancers. And I talked about BRAF, which is a kinase that is abnormal in melanoma, and a drug that inhibits this abnormal kinase has been made, or several actually. Plexicon 4032 is one. It has initially in a patient a phenomenal effect. You can see a patient with many melanoma lesions on the front and the back of his body before treatment, and then a few weeks after treatment, these lesions are all gone. What is really sad is that treatment with this BRAF inhibitor alone is not curative, and almost all of these patients, as you can see in panel C, will unfortunately relapse exactly with lesions in the same place where they were in the first place. But the hope, of course, is the combination of such treatments with additional treatments that may attack other hallmarks of cancer will provide a much, much better outcome. Of course, I can't ignore being at WEHI, venetoclax. So venetoclax is inhibitor of BCL2, which is a recognized driver of chronic lymphocytic leukemia. And these are pictures from the clinical trials done at the Royal Melbourne Hospital by Andrew Roberts, Marianne Anderson, and Jonathan Seymour. You can see a patient with a huge bulk of disease in his abdomen at baseline. And then after 6 and 12 weeks of treatment, a huge reduction in tumor burden. And at the bottom is the data from the clinical trial that led to the FDA approving venetoclax as a treatment for refractory CLL. This is what they call waterfall plots. So anything below the horizon is a good thing. It's a drop in markers of disease like lymphocyte counts in the blood, or the mass of the lymph nodes as determined by CT scanning. So finishing up, so where are we? So in 1971, a very controversial person, Richard Nixon, who obviously was responsible for the Watergate scandal and was, I think, the only US president so far having lost his job as a president, or he actually resigned before he would have been booted out. One good thing he did is he declared the war on cancer and actually increased funding for cancer research in the US by a ridiculous amount of money, and that gave rise to a huge amount of scientific discoveries and following on treatments. So even a person that was quite horrible can do some good things. Maybe something for you to remember. But yeah, where are we? Are we winning the war on cancer? I think we have a lot of new data, of course, since 1971, huge discoveries. In some cancers, there's remarkable success, even with old-fashioned cytotoxic drugs like cyclophosphamide. We now have an arsenal of drugs that are targeted oncogenic drivers like Plexicon for BRAF, venetoclax for BCL2. We need to find ways how to combine them to get the best outcomes for patients of different types of cancerous diseases. On a societal level, we need to find ways to make treatments with these targeted drugs that are insanely expensive, affordable to everyone. We cannot have it that in a society like Australia, only somebody of Kerry Packer's kind of wealth can afford a cancer treatment, and anybody else, well, good luck, try to mortgage your house. At least Australia is doing a hell of a lot of a better job at that through the PBS scheme than the US is doing, for example, where people literally lose their livelihoods with a diagnosis of cancer. Final note, we do have a problem, of course, in terms of cancer. We have a problem with human overpopulation. There's no doubt about that. That will increase the numbers of people with cancer. It's easy maths. We have a problem with increasing pollution and other environmental degradation, and that will lead to more cancers. There's no doubt about it. And so I think if we think about improving cancer prognosis, we should not just think about what we do in the lab. I think we really should think way beyond that, also about societal issues. That's the last point I want to leave you with. So sorry for going over time a little bit. So I'd like to make some general acknowledgments of people that have influenced me and my thinking listed on the left. And I'd like to thank everybody who's worked in our division or my lab over the many, many years, and all collaborators here and beyond. And people who helped me with slides are listed on the right side. Especially I'd like to point out Francois and also Daniel Gray and Eva Michailik for some of the data that I presented. So thank you very much, and I'm looking forward to your questions. And remember, you get Mars bars from Gemma.
Yep, so we'll be taking the questions. Anyone's got a question? Cool. I will just.
You can shout it out, and I'll then say the question again for the people that are online.
Sure. So I just had a question about something you mentioned at the start of your talk with Hodgkin's lymphoma. So you said the transformed cells, the Reed-Sternberg cells, are actually the minority cell subset in the tumor microenvironment, and that's not usually the case, and that's not a common case for a lot of humans. I was just wondering if there was a known explanation as to why these transformed cells are less common.
So the question was about Hodgkin's lymphoma, where the truly malignant, genetically transformed cell is called a Reed-Sternberg cell, but it's actually curiously very, very rare amongst the bulk of the tumor. And the question was, why is that? I don't know. Maybe somebody does. My short answer is, I don't know. But I do know why there is a huge accumulation of reactive cells, and that is these Reed-Sternberg cells. They make bucket loads of many, many different growth factors and cytokines that lymphocytes and myeloid cells absolutely love. So they get drawn in, they get attracted by chemokines, and then they divide, and that then creates the bulk of the disease, which usually sits in a lymph node. And but yeah, the truly malignant transformed cells itself is non-dividing. So it's an aberration, but I love aberrations because I think you can often learn a huge amount from the exception to the rules.
Fresh chocolate's coming your way.
Anybody else got a question?
Yeah, over there. Yeah, just shout it out. I'll call.
Sure. So about aberrations, can you tell us a bit more, other than telomere loss, what are some other ways that lead to genetic instability? I've come across like kinetical attachments and things like that. So, like, I'm sure there's multiple other mechanisms that maybe you can share.
Yeah, so the question was, other than the telomere erosion, what other defects can cause like catastrophic changes to the human genome in a cell? And yeah, you mentioned defects in kinetochore assembly or kinetochore behavior. That's a well-known one. There's many more. I mean, anything that will cause abnormalities in mitotic division of a cell, of course, will cause one of the daughter cells perhaps having one chromosome too many, one chromosome too less in the other daughter cell. It can rip chromosomes apart, and then the DNA doesn't like to be non-attached. So then you get these wrong attachments of where one part of one chromosome becomes attached to another part of another chromosome, and that can be either reciprocal or non-reciprocal. Sometimes you can just lose a huge amount of DNA in one of the daughter cells, and if that amount of DNA happens to contain some tumor suppressors, then, well, of course, that cell will have a competitive advantage. I'm not an expert in what causes catastrophic DNA abnormalities. I hope, I'm sure there'll be some talks from maybe Tony Pappenfuss or others on that. Oh, yeah. Yeah.
Thanks. This is sort of a general question, but so you said at the start one of the ways you could sort of categorize tumors was based on their mutations. But how does this work, I guess, when certain mutations have like such high heterogeneity? Is it sort of like the most common mutations within them that classify the tumors, or?
Yeah, so the question was about classification of tumors within, I guess what you mean, tumors of one tissue type, like breast cancer, dependent on the oncogenic drivers that drives them, and taken into account that within that tumor in the one patient there is large heterogeneity. There are some mutations that are clearly more important than others. And as you said, they're basically decided upon on their frequency. So something like in a breast cancer that is driven by BRCA1 or BRCA2 mutations, you will find that mutation in probably 100% of the malignant cells in that tumor. So you can fairly safely assume that that was pretty critical for the driving the disease and probably sustaining it. And then, yeah, the more prevalent the mutation is, the more it's considered to be an important driver. And if something is there at very low frequency, then, well, maybe it contributes, maybe it's a passenger. But then if that rare mutation in the primary tumor all of a sudden becomes highly prevalent in the metastasis or after therapy, then you've got to change your mind and say, well, that really must matter, because such increases don't happen just by chance. I hope that answered your question. I think, Keshav.
So are there many multicellular organisms that don't develop cancer?
Great question. So the question is, are there multicellular organisms that don't develop cancer? So in the literature, it is said that elephants are highly protected from cancer. But then I don't know how many elephants go to the clinic to be treated. But one thing that is really curious, so elephants have, I think, 24 or 30 copies of the tumor suppressor gene P53. So maybe there is actually something there. I mean, on the complete opposite side of spectrum, there's actually some unicellular organisms, like I think there are mussels in the ocean that can develop cancers, and it's actually a huge problem for the industry that grows them. So they are, and like another really interesting thing that I didn't talk about, those cancers in the mussels are like the Tasmanian devil cancers. They're actually infectious. They're basically transformed cells. Transforming a colony of other cells. So they're really, really interesting. I hope that you're going to get a lecture on the Tasmanian devil cancers as well. I didn't cover it here, but I find that curiosity just absolutely fascinating.
This might be a disaster, okay?
Yeah, don't kill Zilu, please. Yeah.
So I'm wondering if there is any genetic gene therapy target for treating cancer.
Okay, so the question is, are there approaches for gene therapy of cancer? Yes, there have been for quite many years already. Of course, the problem, the hugest problem about that is that you literally would have to get your gene therapy into 100% of the malignant cells. So if your transduction efficiency is even 99%, which would be bloody phenomenal, then the other 1% will just outgrow. But there are efforts nowadays, a lot of them based on CRISPR technology, to try and, for example, you could imagine delete the BCR-ABL fusion gene in CML, or delete the mutated BRAF allele from melanoma. So a lot is being done. I'm a little bit skeptical if this will be possible, but maybe if the delivery will be optimized and it can be combined with several other agents, like classical anti-cancer drugs, maybe targeted anti-cancer drugs, and maybe this could be a huge future achievement. So yeah, I wouldn't throw it out, but yeah, if I would get cancer today, I would be slightly nervous about that approach, whether it will be there to save me.
Yeah, so we've got 5 minutes left, less than 5 minutes, so we will cover one question from Slido. It says, half of all human cancers contain TP53 mutations. Can you comment on the development of drugs that target P53 mutant cancers?
Okay, I hope everybody online heard it, but I'll just briefly repeat it. So the question was, 50% of human cancers and across many diverse types of cancers have mutations in P53. what are the efforts of directly targeting p53 for the treatment of such cancer. So yeah, there's huge amounts of efforts, and actually on both sides of the spectrum. So probably the hugest advance was made the opposite cancers, the ones that have intact p53. So drugs were made that cause an increase in the activity of p53 in the hope that these would then cause the death of these malignant cells. And the best known drugs that do that are called Nutlins, because they were developed at Roche Nutley in New Jersey. And they developed phenomenal drugs. So they're based on inhibiting the major inhibitor of p53, which is called MDM2. It's an E3 ubiquitin ligase. So they work phenomenally. There's one problem. They are so toxic to many essential normal cells in the human body that their progression in clinical trials is going extremely, extremely poorly. So they were mostly trialed in AML, and they completely wipe out the AML cells in the patient, but they also wipe out the normal stem cells in the bone marrow. So the only setting that this drug can be used is actually a non-genotoxic clearance and preparation for hematopoietic stem cell transplant. And that's been done in a very small number of patients. It's been spectacularly successful, because the advantage there is that you clear the bone marrow not by a DNA damaging drug that then mutates other cells, but a non-genotoxic treatment. Like many drugs that are highly effective but highly toxic, I mean, maybe the future will be if they can be combined with an antibody that targets them to the cancer cells and away from the normal cells. Maybe there's a future. So the other side of the question, yet targeting mutant p53, there have been compounds that have been published to do that, but the evidence for that is actually very poor, that this is the mechanism of action. So much more needs to be done in that setting. Whether that will be feasible will depend on whether this gain of function effect is actually really essential for sustained expansion of tumor. And you can talk to Zilu about that. She will have a very strong opinion on that. I'll leave it at that.
Oh, sorry. Yeah, just out of my own sort of, I do have one last short question. With just now you're saying that evading cell death can that potentially not lead to cancer, right? And then you showed us one experiment where you have a normal cell and then you add radiation that gives pressure to the stem cells, right? And so that gives a higher likelihood. And then you mutate one of the initiators of the BCL2 protein family, and then you have radiation, then there's no cancer. But then the point is, would radiation potentially help to suppress some of the tumor generation? So say if you don't have radiation, you just have the initiator being activated, and then you wait for a while, would that have any impact at all?
I think people have been thinking about things like that and assuming that everybody who's 50 years and beyond probably has a certain number of abnormal cells that, if things go badly, will cause a cancer in that person 10 or 20 years later. And so there have been experiments along these lines. I think the most interesting experiments I know of in mouse models is where they had a transgenic system where they could increase the activity of p53 in a regulated manner, and where they would, every few months in a pulsatile manner, increase p53 activity and then bring it down to normal again. And in certain cancer models, that actually prolonged lifespan of the mice. So maybe that's a way to go. But yeah, initially it was treated by things like radiation, but tried with radiation, at least in animal models. I don't think it's been done in humans.
Okay, thanks, Andreas, for the seminar. I think we'll call it the end for the moment. Yeah, thanks.
Thanks for having me.